Welcome to everybody out there who is joining us for this deep dive webinar in understanding the microbiome of your brewery, Metagenomics 101. This is brought to you by BioMaru, and warm welcome from them. They're a global leader in beverage quality diagnostics. If you are in the brewing world, you know the products that they make, and what we're going to talk about is what they understand about that microbiome within your brewery and ways that you as a brewer can more effectively understand it yourself. In today's brewing world, there is nothing worse than that kind of microbiome getting out of control and doing things in your brew house, in your fermentation cellar that you don't expect it to do. Fixing mistakes is a costly thing. And in this day and age, with the way that costs have increased across the board in the economy, we want everyone to be operating at the most efficient way. And so joining us for this deep dive webinar, Dr. Daryl Barkhouse. Welcome, Daryl.
Hi. How you doing?
Good. Daryl, why don't you tell us a little bit about BioMaru and give us a little bit of background, on your history, both in this kind of world of microbiology as well as brewing, because yours spans a little bit of both.
Yeah. So in terms of the company at BioMaru, our founding individual goes back to the days of Pasteur. So he was a direct student of Pasteur and created the company after those times of studying with Pasteur. So we have a long history of microbiology, as you can imagine. And then with BioMaru, and when we were previously Invisible Sentinel, we have a very large footprint in the brewing industry of really partnering with our craft beer and then also larger beer manufacturers to make PCR test kits that were really correct for the business and directly what the craft brewers and other brewers needed for their quality programs. For me, I started my career in the brewing industry, so I always love getting to talk to anyone in the brewing industry. And it can I can go off in tangents quite a bit, so please keep me in check. But brewing is close to my heart because it's where I started. I was the brewing microbiologist at the largest brewery in Toronto for Molson Breweries, and that's where I started. And I was also responsible for propagating yeast for 8 breweries, which included all Molson and then Moosehead. If anyone's familiar with Canada, they might know Moosehead beer. After leaving Molson, I did a stint in pharma. Then I went back and got a couple more degrees. Then I took a job with Invisible Sentinel, which was started by a former classmate of mine. And it was a perfect fit because I had a lot of quantitative PCR experience and brewing experience. So started making PCR test kits and assays for the brewing community. After that, I started the genomics facility that we have now. So it's been growing for about the last 6 years. And now here we are today to talk about it.
And so our goal for this session is to understand the fundamentals of metagenomics. Obviously, there's this unseen biological world. You shouldn't say unseen. You make the tools to make that unseen seen, don't you?
Yeah, that's correct. The biggest issue, is what's lurking that we can't see, that we can't grow on our plates or can't see in chemistry or can't detect in sensory, or we detect when it's too late.
So we're going to talk about those methods of, troubleshooting contamination, and understanding the way that, this microbiome works across, that entire scope of the brew house. And we're also going to talk about the, various tools that brewers can use, including some of these more advanced service-based tools to gain the deepest kind of understanding. And that's basically what metagenomics ultimately comes back to. But maybe we start first, Daryl, by talking a little bit about what that brewery microbiome is. Why don't you kind of give us a background on what we're talking about when we're talking about microbiome?
Sure. So when we're talking about microbiome just really means all of the microorganisms in some defined area. So it could be the entire earth, it could be a river. If you follow science in the news, you hear a lot about the human gut microbiome, how that affects neurological diseases. Soil microbiome, we're talking about the environment, looking for diversity, impact of things on the environment, that kind of thing. And then for us here, we're talking about in the brewery. So it could be in your whole brewery, it could be in your yeast springs, it could be in your fermentation vessels. So whatever defined area you have, it just means all the microorganisms that are in that space.
Sure. And we take the value off of this. It's not like a microbiome is a good or a bad thing. It is simply this kind of complex environment that we're in, with various microorganisms all competing in this natural selection kind of form for their own survival and doing the things that they can, taking advantage of circumstances that arise to them. And then, of course, those things we don't want out there are ones we try to control you and understand, but we can't really control what we don't know is there, right?
Exactly, exactly. When I was doing my PhD, a lab had a great sign outside. It was a radiology lab, and they said, if you can't see it, you can't treat it. So I always thought that was a good explanation for this kind of microbiology as well. If you can't see it, you can't figure out what the issue is. You can't ID it.
So you have this microbiome within the brewery, but then you're, in a typical brewery process, brewers are always constantly bringing new inputs into that environment, right? You bring in new grain, you bring in a new yeast pitch, you bring in hops, and so you're constantly also altering and changing, potentially bringing in new variables into that microbiome and potentially introducing new challenges, new players into this. Talk a little bit about how all of these inputs ultimately impact that microbiome.
Yeah, I mean, you're definitely right. I mean, this industry is always evolving so much as we've seen, the last 20, 25 years. There's always, new flavors. You want some kind of fruit and sour or guava sweetener or, lactose beer. So all these kind of more, unusual adjuncts you bring in or processes like, heavy dry hopping on the cold side. All of these things can introduce things that we're not generally used to looking for. And our media hasn't been designed to find these things, and we haven't been programmed to design it. But then also, our products are evolving, right? Lower bitterness, lower alcohol, all the things that have helped us for, thousands of years keep beer from spoiling. We're kind of taking away. It's similar to the food industry where we're taking out salt and sugar. So now things are growing in our ketchup and soup. So it's kind of the similar thing. As our products evolve, so too do the niches that these microorganisms can grow in. So sometimes we're creating these niches that are perfect for organisms we're not familiar with.
Killing our hops at a lower temperature in order to maintain more of the oils, but then we're not
Yeah.
denaturing enzymes, and
Yeah.
who knows what else is coming in then,
Yeah.
with it. Some of the things that we do for other reasons in a brewery can have some unintended effects, and especially in that kind of scope. But anyway, so let's talk about some of some of these kind of like general ways that brewers can start to get an idea of what actually is going on in that microbiome in the brewery. We definitely have talked here about what, like that need for some monitoring. I think every brewer out there knows they need to know if there are things in those beers, or in their entire environment somehow that are causing some challenges. So let's talk about some of these various methods that brewers can use to gain some understanding of the microbiome itself.
Yeah, definitely. And I just want to start off by saying that, we view metagenomics and sequencing and these kind of higher level technologies kind of as an enhancement or a supplement to what we currently do. This isn't going to replace plating tomorrow, right? Maybe in 10 or 20 years, sequencing might come down to that level. But for now, we still need plating, we still need PCR, and we can add sequencing to solve different issues that arrive.
One,
So,
there, I am. When you say sequencing, I admit I'm a little bit lost here. So why don't you explain to me, just what we're talking about when we, I think brewers are familiar with plating, right? Using media. They're generally familiar with PCR, but maybe you can give a little overview of what those are, and
yeah,
then maybe describe what the actual process of sequencing and metagenomics is.
okay. Yeah, so I think we're all familiar with plating. Plating is cheap. We're generally familiar with it. It gives you a decent idea of what's happening in your brewery. The drawback to plating is it can take a long time, especially for anaerobic cultures. You might have to take 2 weeks for some weird PDO to grow up, and all that time you kind of don't know what's going on. Logistics is wanting to clear the warehouse, all of these things. So things are piling up. So plating can suffer from that, and also it's estimated about 98 to 99% of what grows in a manufacturing environment cannot be grown on standard media. Um, and it's the truth. So just the things that are out there that we don't know about, we can't grow them. And so we're missing them. So you can get false negatives. This is how, you get products spoiling out in the market, but your plating was all clean for that week or that run or, that month. So plating does have drawbacks in that area. I think
And then you've got this
sorry,
specificity to media, right? That plates can only grow
yes.
certain things, and so now in order to find all of the different things, like you're not making lots and lots of plates for stuff.
Yeah, yeah. And I think everyone knows, like, which LAB plate do I pick? Like, do I pick all six? Do I pick three of them? Do I pick two? And that just tells you that, not any one of them is really great at capturing everything. Okay. So when we think about PCR, the inclusivity and exclusivity on PCR is very, very good, right? And that's because we're targeting specific either species or genera. So although the specificity is really good and we're excluding everything that is not that target, it's a very small target group, right? So your plate, you might say, okay, it's lactic acid bacteria on here, but your PCR is going to be tighter than that. So you're not going to be able to see as much. So you have to know your target, and that's all you're going to find with it.
When we say PCR, what is the technology behind PCR?
Okay, so yeah, so if people are familiar with the idea of primers, so primers are small DNA segments that target a specific area of the genome. So if you want to look at Lactobacillus brevis, you look at all the data that's out there, or in our case, if there's not enough data, then we collect everybody's strains from all of our partners and sequence it so we have the data. Then you say, of all of the Lactobacillus out there, here is one spot that's unique. And if this area of the genome is there, I know it's Lactobacillus brevis. So we make primers, which are little targeting strands of DNA that go on either side of that sequence. And then, so that's when it's in our test kits. It consists of primers, then the polymerase that recognizes when those primers bind to the target DNA. So if that brevis is there, the targeting primers are going to bind to its DNA. And then the polymerase is going to make copies of it. So now, like, no one can really see one copy of a genome or of a target sequence. But once you do polymerase chain reaction, then you get 10 billion copies. And then that's very easy to identify. And then we use some fluorescent light to identify and say, yes, hey, look, there's 1 billion or 10 billion copies of this. So we know it's this species.
So that's a bit like listening to the first 4 seconds of the song and being able to identify the song without hearing the whole song.
Yes,
which you can,
exactly.
I mean, I love those quizzes, right? Right.
Yes.
And then, and then explain, what sequencing and metagenomics is, as an expansion from that.
Yeah. So as a difference to PCR, where you're targeting this one small area of DNA for a very specific purpose, for a very specific organism. When we talk about metagenomics, we're talking about sequencing everything that's there. So it's agnostic. We don't have to grow it up on a plate. We don't have to have an assay kit for it that is specific for it. It's agnostic. We say, here's a sample. It has a bunch of microorganisms in it. Tell us what they are. So that's what we do. So we take all the microorganisms that are there, smash them open, get their DNA, and sequence it. Now, there are different levels of sequencing with different resolution. It could be shotgun, where you're sequencing all of the genes of every microorganism in your sample. Or it could be something that people are probably more familiar with, which would be 16S or ITS metagenomic sequencing. And that's when you just target 1 or 2 areas of the ribosomal gene from bacteria for 16S and from fungi for ITS. So with one, you get the whole big picture, and with one, you get a smaller picture, but it's agnostic and you identify everything in your sample.
That's an interesting way to look at this then, and that kind of speaks to this resolution idea that, if you know exactly what you're trying to find or not find, plating, is a way to get that. Obviously, there's some time challenges to that. And often within a brewery environment, like people, brewers want to move things before they might have an actual answer because tank time is money. And, and so it can be good looking back and you can decide that, something's going on there. Before you package it, but it's, it will have taken a few steps further in the brewery for that PCR, right? That gives you a little faster, kind of answer on some of that.
Yes. So that's the thing. Plating has, decent resolution, but takes more time. PCR is very fine resolution and very fast. A lot of our assays are done just from a sample without enrichment. So you can have, same half shift results. Some require a little bit of enrichment, a day or two, but still much faster than plating. Metagenomics can take a little bit longer. It's kind of in that range of plating, but the data that you get from it and the resolution of that data allows you to make way deeper decisions about what's going on in your brewery and then what corrective action steps to take. And I'll just explain resolution too. Let's say you have an HLP plate. There's some growth on it, so you say, okay, it's a lacto or PDO. It's a lacto. And that's what you get. So you kind of know the genus maybe, but now the way they've split up lactobacillus, it could be like any of 27 genera now. Anyway, so PCR, you might say, okay, I have a PCR kit for hop-resistant lactobacillus. So if you get a hit on that, a little more, right? It's got a little more risk to it. You want to put it in your low bitterness beer, that kind of thing. Now, if you use shotgun sequencing, you might be able to get an entire picture of the genome. So you could say, oh, look, this is the lactobacillus that I purchased 3 years ago from a supplier because I see its signature. So hey, we did this to ourselves if it's a contamination problem. So that's just kind of an example of the resolution of the different technologies and what it can tell you.
Sure. And I should also note, if folks are following in out there live while we're doing this, throw some, if you've got any comments, if you've got any questions, if you need some more clarification for what we're going through, just toss a comment up there. We'll keep an eye on it and I'll try to throw those in there where they make sense. But we are here to answer questions and help you understand the microbiome of your brewery. And, and so that's the whole point. So yeah, feel free to jump in there. And then in terms of cost, there definitely is a scale here. But this is not necessarily the kind of thing you decide on from a brewery perspective based on what your budget is. You really approach it based on what you are trying to accomplish. And plating and PCR can give you answers to very, very specific questions, whereas that sequencing and metagenomics approach can help you, it's almost like, it's a bigger map rather than, just, an answer to a specific question.
Yeah, that's correct. And, we're never going to recommend that the only thing you do in your brewery is sequencing. Kind of that would be madness. And, that's not necessary and it's not tenable. Where we see sequencing is when you have those problems that can't be solved. There's some known unknowns or unknown unknowns that are holding you back. And in science, we always say there's never time to do it correctly, but there's always time to repeat it. It's the same kind of thing. There's never budget to do sequencing, but then there's always a budget for a recall. So it just depends on what how much money you want to spend and where you want to spend it. So yeah, there's a place for all of these technologies. Plating is probably not going away. PCR will get more and more. And now sequencing will be, brought in and used as that higher level tool to help you solve bigger picture problems and problems with those known unknowns and unknown unknowns.
Because this isn't just about the presence necessarily of a single thing. There's also a time factor involved here, right? As beer moves through a process from hot side going through a chiller, getting through knockout. Maybe it goes through a hop back or gets hop dipped through a flowable hop product now in a like preheat export. We have all sorts of new fun tools for making beer in creative ways. And so things, there are plenty of these opportunities for things to jump in there. But then there's also this question of some things that can actually have some effect on flavor within a beer may also not be alive at the time, or the alcohol may very quickly kill them in fermentation. And so you have some question as to some of the methods that can or can't find some of these things that can have an effect, but they may again not be responsive to some other testing methods. Talk to me a little bit about that.
Yeah, you are correct. You're kind of bringing the concept of what some people say is like dead DNA, things that were there but they've died off in the process, just like you said. So if you can't grow them, they're not viable. You can't grow them up, so you don't have evidence. With sequencing, depending on how you do it, you can capture that DNA. You can even get DNA if you're pasteurizing coming out the other end of the pasteurizer, so you can still capture it at that time. And these things are usually still cell associated, so it's usually pretty easy to just spin them out of a solution. So it's not generally just DNA floating around in liquid. It's generally still cell associated, even though those cells are beat up a little bit. So it can help give you a picture of what happened throughout the process. Yeah.
And this beer sense, I think back to that study that they did a number of years ago on lambic, trying to understand some of the, because like, well, we're testing, this as it gets through it just to see what's in it. And, oh, it's only these things. But, as they went back and started testing every single day from day
Yeah.
1, day 2, day 3, then all of a sudden you find out, oh, the first 3 days you got butyric acid bacteria
Yeah.
that are actually making some of these things, and they are dead after day 3 or day 2.
Yeah.
I can't remember the exact dates on that, but they're actually creating flavor precursors that then other bacteria we're going to work on, or wild yeast are going to take those things. And so if you only are looking at it at one point, during that fermentation or post fermentation process, like it's never the full picture. And because these things can set up some of these, these things all along the way there.
Yeah, exactly. Like one snapshot is very often not enough to tell you about the full evolution of a product. You're definitely correct. So we can do that. And then, that also brings up the question, well, what if we don't want the dead DNA? All we want is the live DNA because we want to know what's going to happen when it's in market, that kind of thing. And then that's doable as well. So there are ways to clean out the dead DNA out of our assays. And, some of our partners are using that technology right now. We can use the same technology that we sell or provide for PCR kits. That's also usable in the sequencing industry.
Sure. Well, let's talk a little bit then, about how metagenomics works in practice, and what the actual process is of what the actual process is of using the, this metagenomics approach to understanding what's happening at various points along the process.
All right. Yeah, I mean, we try and make it simple, and those of you in the brewery are kind of already doing a good portion of this. Sample collection is basically the same as what you do now. We need nice aseptic samples. So the same way that you want your plating results to be a reflection of what's actually growing and not a dirty spigot, it's the same thing for metagenomics. You want that nice aseptic sample. We're generally using the same kind of sampling devices, World Pack bags, conicals. If you need to ship it to us to do this work, just ask me for recommendations. Some things leak a little more than others, and we don't want things leaking. So it's a little different than going from the floor, walking back up to the lab, than it is, having it thrown around a FedEx truck. So that's the only difference, is we sometimes use a little bit better sampling devices. But same collection, you'll take it from wherever, we're going to rely on your expertise as well as ours to figure out where samples should be taken, all of that stuff. But we're always here ready to consult. Once you're ready, you ship them out in the mail to us. We really like FedEx. I really do. They give us the least problems. Everything's a little problematic post COVID, but FedEx for me works the best in Philly. Yeah, and the shipping is pretty easy. If you, for some reason, had something that needed to be frozen, you would ship it on dry ice. We can help with that. If it's fermenter sample and you don't want it overgrowing, then ship it to us cold on ice. If it's cans of finished product that's going to be in the market, then you can just ship it to us at room temp. It's pretty basic. So all that first half is the exact same things that you do every day in the brewery, and then once we get it in our hands, then we do kind of the higher level work and make it easy on you. So I mentioned that most of the the DNA we'll be looking at is cell associated. So it's in the bacteria, it's in the yeast, it's in the mold if for some reason there might be some mold around. So we use some different techniques depending on what the samples are to smash open the cells. We use physical and chemical lysis to break them open. And then we clean away all the debris. So get rid of all that cellular debris, all the plasma membranes, all that stuff. So we have nice, clean DNA. Now, we use largely Illumina sequencing, which is referred to as short read. Some people may have heard the term Oxford Nanopore. We do a little bit of Oxford Nanopore, and bioMérieux does own a substantial share of Oxford Nanopore, so I just want to disclose that. But 99% of what's out there is Illumina sequencing, which is short read. What that means is you take all that DNA that we cleaned up, and it's beautiful for us to use, and we chop it up into 600 base pair pieces. Then we put some barcodes on the end of it so that we know, hey, this piece came from sample 1, this piece came from sample 2, and so on, because you're generally not running one sample. That would be a little inefficient and wouldn't tell you a whole lot. So we barcode them, And then we add some adapters to them. The adapters allow that to stick to the flow cell, and then that flow cell is where the sequencing happens. So sequencing is basically just repeated PCR with an output that measures, hey, I added a C or I added a T or an A or a G at this point, and it builds the sequence that way. It's called sequencing by synthesis. The reason it's about 600 max with the older platforms is that's about how long the polymerase can be reliable. With the new Illumina platform that was just released this year, they're looking at 1,000 base pair. So now you can start getting in better resolution and start kind of eating into that Oxford nanopore space of longer reads. So Illumina is constantly evolving and trying to get longer and longer reads. And that's it. So once we have the reads that say, okay, these are the 600 base pair sequences, we use a fancy computer to stitch them all back together and create genomes. Then once we have those genomes created, then we use other software that says, hey, these are all the organisms that you have there, and you can look at relative abundance, that kind of thing, if that's important to you as well. And that's pretty much it. And then you get a nice report from us, and then we start troubleshooting or figuring out corrective actions if that's what you need.
Let's talk about some of those kinds of applications then, in terms of what, something like abundance means, in a brewing space, and, what some of those kinds of common things that you tend to find through this kind of, this search process. From a broad kind of perspective, what do most breweries come to you with? What's the most common issue that they are trying to solve for when they want to pursue this kind of metagenomics approach?
Yeah, it's really pretty basic, and overall idea is that, hey, we've done all of our plating, we sent stuff out to a third-party lab, they couldn't find it. We know there's growth in here because we see it, it's cloudy, or it's giving us sulfur or whatever, and they just can't identify it with traditional methods. And if you can't identify it, then there's no way to track it down to a biofilm or a pump or a raw material. So it's really just IDing stuff where traditional methods have not been capable and haven't had the resolution they need. That's the most common, and that's across all industries, honestly.
Sure. And we shouldn't only cast this in the kind of realm of problem solving. Ideally speaking, in a perfect world, we would be doing preventative or research to understand what that microbiome is so that we can proactively adjust for that and not simply have to solve and reverse engineer a problem that occurred and get to the bottom of it. There is some preventative approach. I mean, we all know that preventative healthcare is the smartest way, the cheapest way, ultimately, right? Much less expensive than the kind of traumatic care, emergency care that it takes. And yet, in this strange kind of funny way, we seem to you can always find a budget to solve a major expensive problem like that. It's harder to find a budget to get that preventative care up and running in order to understand it. But in this sense, that kind of understanding what's going on in your brewery microbiome before there's a problem can also be useful.
Yeah, and again, same common across all industries, right? It's always difficult to get a preventative budget if you have never had a problem. Where we see, the biggest kind of adoption of these kind of protocols is after someone's had a recall or brand damage. Because then people realize, the ROI you can get with this, especially if you have, reoccurring issues, recalls, reoccurring brand damage. None of those things are great. So if you can get ownership or leadership on board with preventive without an issue having popped up, then that's great, and more power to you. It is difficult, and it's not, it's understandable. That's the way business works, and it is across all industries. But it is very helpful, and we'll talk. I can talk a little bit about that kind of preventive monitoring in a later slide here.
Sure. When I think about this, and when I think about the kind of impact of metagenomics, I think about something like that, the Bourbon County Stout issue with lactobacillus acetotolerans back in, I think it was 2015 when that hit. And all of a sudden, an organism that they didn't know could exist in that high alcohol environment ultimately ended up spoiling significant amounts of that batch, created an entire recall a very, very expensive problem that they had to solve for. But of course, and that acetotolerance did not grow on any of the media that they're using to test with, and so all of these tested clean using typical methods, and yet there was still this extraordinarily expensive recall at the end of it, because you take all those barrels, you put them in much larger blending tanks, and then all of a sudden, whatever might have spoiled one of those barrels,
Yeah.
55-gallon spirits barrels, is now spoiling an entire 100-barrel or 100 barrel tank that they're blending in. Like,
Yeah.
all of a sudden you got a major problem on your hands. But something like metagenomics is designed to help you understand all of the other things that you might not even know that you were necessarily looking for at the time.
Yeah, correct. That acetotolerance, it kind of falls somewhere, depending how much insight you had at the time, but between a known unknown and an unknown unknown, and that's how they come up and kind of bite you, and cause those issues. And the other thing with the craft industry, in the pursuit of these amazing kind of artisanal products, we're creating these niches, right? So they created it. We're always, you alluded to it earlier, we're always creating new niches. And so metagenomics is a way for us to keep on top of that. If you're going to bring in a new product to your brewery, say you might going to co-pack or produce for someone else, or just change up a product to something you're not used to, it's always a good idea to do a survey of your area, see what's lurking there, and then understand that industry, because there could be things that have never affected your brewery, and they might even be house character for your beer, but then you change up your product, or you bring in another person's product, and it will completely ruin the flavor. So metagenomics is a really good way to get ahead of it, so that you can validate that process is going to work and not cause you headaches on the back end. So it's a very good way to get ahead of those things and kind of be visionary.
In terms of places in the brew house and then the cellar that typically become problem inducing, or I should say environments that harbor some potential or have a higher prevalence of harboring some of these challenging microorganisms, from your perspective, what are some of the major culprits that you've seen over the years?
Yeah. So, depending on the size, of your manufacturing facility, of your brew house, it could just be common CIP shadows, from your manhole entry points. If you don't have someone jumping in there and hand scrubbing them with, a green scrubby, that could always be a point. You may have, if it's an older facility or if you've done work, you may have dead legs, and you're like, okay, well, that dead leg is shorter than 2 times the diameter, so it's not a problem. Till you go and look at it and you're like, oh, there is a biofilm in there. It's a problem. The math says it's not, but the microbiology says it is. We see things that you can only find them in drains and never find them anywhere else in the brewery, but they end up in the product. So, it's really, really dependent on the facility and just how the operation runs. But it's all the common things. It can be flexible hoses if you use it. It can be dead legs. I had issues where we were kind of victim of PLC brewing. The PLC said the solenoid opened, so it opened. But when you go and put your hand on it, there's no click. So there's all of these things, and metagenomics can help you kind of validate and verify those things. So anywhere there's a process that's supposed to work, or a pipe that's supposed to work right, there are things that can fail, and it can harbor microbes.
What is biofilm? You've used that term a few times. I just I'm curious and also, what is a dead leg? When you say that, what do you mean?
Oh, so like a pipe that doesn't flow, it comes to a dead end, and so you get weird eddies and stuff like that, and you don't get a full CIP. The CIP can't.
Oh, it might. So it's like
through it.
a T where you've blocked the T off or something, and then now
Yeah,
you've got this little
yeah,
cavity along rather than that smooth pipe for the
yeah.
CIP.
Yeah, so if you've never had dead legs, then you are seriously lucky. I applaud you, because they can be a nightmare.
But then biofilm, just
Yes.
what is biofilm?
So one biofilm is really, it's a kind of a layer of microbes where they've protected themselves. So they've produced some kind of glycoprotein or proteoglycan or something where it forms a protective layer to protect it from your CIP process and your sanitization. And then the really tricky thing about biofilms is very often it takes very, very few microorganisms to create that biofilm. So you can just have these tiny amounts sloughing off over time that you're never going to catch with your plating. And that can sometimes be difficult even with sequencing. And so you might use an enrichment protocol there. But they're really things that are kind of protecting themselves from your CIP. And then so when they're protected from CIP, you can't sanitize against them. So that's really it. And they can be low numbers and they're very hard to detect.
We had a question come in, about the impact of sequencing in the NA space. In the alcoholic beer space, you have this added layer of protection in that alcohol will kill a lot of these or any of the uh, potential pathogens that could harm human health.
Yeah.
That doesn't mean that's going to protect from off flavors and things that might cause product recalls. But then we start talking about the NA space, and the stakes get even higher, right? Because without alcohol to provide that kind of protective layer, it needs that packaged NA beer has to be pristine in a microbial sense.
Yeah, you're right. It's a very large added risk. I was lucky when I worked, in the microbrewery space, we made 1.5% product and we pasteurized it, to death. So, it wasn't that big a deal for me. It didn't become a headache, but in the NA space now, it's booming and there's a lot more risk there. So metagenomics can help you kind of identify if any of those risks are there. And then, once you know what might be there, you can also look at processes and probiotics and these kind of things. I mean, brewing yeast is a probiotic, right? By definition, because you use it to suppress everything else in the fermenter, right? That's why you want rapid growth at the start. So once you know what's there, then you can, work up your corrective actions or your action plan on brewing. But if you don't know, you're kind of always going to be shipping your stuff at risk if you're not, sterile filtering or something.
Another potential application for this is especially understanding what we're seeing more and more brewers, especially like even in the IPA space right now. I can tell you off the top of my head a number of brewers who are blending yeasts, especially in hazy IPA, to accomplish different kinds of flavor goals. That whether that's thiol expression, whether that's haze stability, whether that's beautiful kind of peachy stone fruit character, whatever that is. I hear, and there are some that are blending in three-way kind of yeast triads. And of
Yeah.
course, understanding then how those blends of yeast stable or don't stay stable over time can be an important use of this. Again, you're not just talking about presence, non-presence. You can start talking about concentration and get an idea through this as to which yeasts in a kind of blend of sorts are out competing each other, and which ones are doing the heavy lifting, which ones are thriving in that environment, and which ones might not be thriving.
Yeah, that is a really good point. When you're looking at yeast blends, like you said, is happening more often now. You kind of have to rely right now on sensory. You say, okay, after we repitch this 6 times, our sensory starts to get off target. So we know, we can recollect this 5 times and use it. But you don't really have like kind of better hard data to go on or know, why is the sensory off? Is one thing outgrowing the others? So if you use metagenomics and you have signatures for each of those yeasts, you can track over time. So you can look at your Gen 0, your Gen 1, 2, 3, 4, however many pitches you want to go and say, look, this is why the flavor's off. And so, it's theoretically possible that you can repitch some elements. If you say, okay, component 1 is kind of outgrowing 3, then when you recollect on the 4th thing, you may have had like a prop tank with a small amount of number 3 in it, and you repitch and mix that so you can keep going. If that's more economical than starting over, which, I would think it very well could be. So yeah, definitely, that is a good use of it.
Yeah, let's not throw out expensive beers that you've
Yeah.
thrown a whole bunch of Whirlpool hops in on, and
Yeah.
and certainly not that you've started dry hopping, because then you're just flushing money
Yeah.
down the drain at
Yeah.
that point.
Nobody wants that.
Sure, sure. Well, let's talk then about the kind of benefit, because this is this system, metagenomics, is built to work within existing brewery quality systems and quality departments.
Yeah, definitely. So like I said, this is not a replacement technology. We're not going to be replacing plating. We're not going to be, usurping the space of PCR. Those things have definite big advantages. We want to supplement or enhance your quality program. with metagenomics. It gives you, that ability to detect the whole range of contaminants are there. You don't need to target it. You can find those, if you've heard the term VBNC, viable but non-culturable, those would be your known unknowns. So all of that you can find, right? So you get a really good picture of the complex microbiome of your brewery, which is a lot, more than you can get with plating and PCR. You can get improved traceability, right? If you can say, what I found here in my product today, I found it 2 weeks ago, in my raw material, then you can start making, stronger partnerships with your supplier. You can go back and say, hey, look, like this isn't on the C of A, but we're finding it. So can we work this out? And, can we help you make a better product so we can make a better product? If it's, a little more insidious than that, you can say, hey, look, this guava you gave us was full of this bacteria. It destroyed things. Can we get a little money back? Can we change the process? So those are the kind of
you're
things.
saying that aseptic puree might not be as aseptic as they claim from time to time.
Yes, it's as aseptic as the C of A says it is, right? Just about.
Sure.
Yeah. Okay. Yeah. And then, cost savings. This is a big one, right? Because if you're trying to explain to your leadership what the ROI on this is, you got to make a big case because, it's not a $2 plate and it's not whatever you pay for a PCR assay, which I should know, but I don't because I'm not in sales. It's not $20 or $40 or $10. So you really got to make your case and show that ROI. And it really is the tool that gives you that bigger picture. So when you have a small problem arise, you can get your corrective actions, figure out your game plan, correct it so it doesn't turn into a bigger problem over and over again. And that's really one of the great qualities of metagenomics and sequencing is really giving you that big picture and be preventative and have that insight.
And this is a good technology potentially to use when validating, from new providers or bringing new sources of ingredients into the brew house. Try to understand what that microbiome is, in addition, and how that might impact your particular microbiome.
Yeah, for sure. Like I said, anything that you're bringing into your brewery can affect everything else that's there, good or bad. So yeah, knowing it, like I said, if you're bringing in new products or you're creating, different wort streams or hops or adjuncts, this is really a way to validate ahead of time that you're not going to be seeing any problems because you really want to know. Or if you can, create problems, you want to know and be prepared to solve them ahead of time. So, just because you see something pop up that's bad doesn't mean that you can't do it. Just means you got to have a game plan. One other nice area that I like it, and especially on ROI, because it's cost savings, is let's say you have a problem, you can't pinpoint it. Like, what do? You bomb the whole brewery, right? If it's a big problem, you just bomb it because you just bomb it and hope you're going to get what you needed. If you're using metagenomics, you can help pinpoint so that you're not shutting your whole brewery down for 3 days or 4 days or a shift. You're saying, hey, look, we have to, close off these 4 vessels or close off this kettle or whatever. You can pinpoint things. So you do targeted remediation without shutting down your whole operation. And then also just wasting all those chemicals, right, that you're using for your CIP if you can do targeted. So you can get a really big ROI that way when you have to go and clean up after an event.
And it goes without saying that the cost of this tends to be far, far exponentially less expensive than product recalls, not just in terms of replacing or refunding that product, but also potentially the damage to brand that can come from some of these high-profile recalls that you've seen out there. I mean, I think we live in this this trust environment, right, where brewers are making the most high trust product out there because it's something that people put inside of their bodies. There's nothing more intimate than something people put inside of themselves. And so having that trust in the quality of that production is like, it's so much more important even, than the whole cost issue, at play here with recall. The idea that people may not trust you as a brand to make a high quality product that's going to be safe or correct for them. Once that trust is gone, that is the most expensive thing, to potentially rebuild with consumers.
Yeah, definitely. It's such an important relationship. And I mean, you've been in this industry a long time. When you talk with people in the brewing industry, you can tell that we make products that our friends and family will consume. And so, I take it personally when something goes wrong, and I think most people do because they try so hard to make a great product that they feel safe inviting their friends and family to drink. And so, it's difficult when things go wrong. So this is a really nice tool to give you even more confidence so that the products you're producing are safe and you can continue to, build that relationship and not have damage done to it because it is the most important one. You're right.
Then, another thing that this can be used for is understanding, say water in the brew house, right? Folks using water sources, over the for the variety of potential inputs, yeah, or water sources, public water sources, well water sources, wherever they're understanding what's in this raw ingredient that you bring in. This
Yep.
is, you can apply that to with this process.
Yeah, definitely. This outside of, doing the PCA plates or some coliforms, we generally don't do a whole lot, of micro on water, do a little bit of chemistry. But there are so many things that can change, right? Just the time of year change. If you're not on city water, if you're on wells or whatever, but even if you're on city water, the time of year can change. You get water main breaks that allow big slugs of soil in. If you're in wells, like things change, pHs change, the amount of clay changes. All of these things can affect the microbes. You can have, metallo loving organisms that start to grow different times a year. You can have thermophiles, right? If you have hot water processes, you can create a niche for thermophiles. We're not generally looking for any of these things with standard media. So if you're seeing problems, that you think is in the water, but you can't tell, then it's a good place to look, and it's a good place to use metagenomics to solve it.
Thermophiles. I'll tell you, my microbiology vocabulary is growing by leaps and bounds through this conversation. Well, maybe
Yeah,
let's
stuff that makes it through your pasteurizer.
and we can get into like nerdy microbiome questions, in a minute here because it does sound like some folks do truly want to get in the insane weeds with you on this. But say a brewery out there that's watching or listening to this wants to get involved with metagenomics. Talk about the kind of investment that you guys have made, and what that means in a practical sense, and how what you all have now built in order to provide this service and this partnership with a larger number of breweries.
Yeah, sure. So we were formerly in University City. If anyone's familiar with Drexel or Penn, we were nestled right in between those two. And then last year we built a three times larger facility in Deep South Philly in the Navy Yard, right next to the Super Bowl champion Eagles Field. So it's a great place to be, but it's difficult to get in and out when there's a game. I'll tell you that. It's a good day to work from home when that happens. So we have a much bigger footprint. My lab space is
I,
about
I don't want to
five
mess.
times as big.
We're not going to mess with those Eagles fans, okay?
Yeah,
We're
it's
not.
not a good
No,
idea. I
no,
wouldn't
no,
either.
not going to mess with them at all.
Yeah, yeah. And I'm not native here, so I stay out of it. Okay, yeah. So in our facility, so we do metagenomics. We do shotgun metagenomics. We also do whole genome sequencing, which there could be some applications for in brewing. If if you want to know, hey, this is my exact yeast I started with 100 years ago. We could tell you that. When I was at Molson, we had those claims, so it would have been interesting then if I had this technology. The other thing is, so if you come to us with an issue and we don't have another assay, or the industry doesn't have an assay for it, we can partner with you to make you a custom PCR assay. That's a big portion of what we do, and that R&D lab is adjacent to mine, and it's much bigger than my lab. There's a lot of great scientists in there. So at this location, we're really focused on being partners with industry. It's the only way that we can really serve people. Like, we don't want to be an assay or kit supplier. We really want to be a kind of a solutions partner to work out problems together. So that's what all the people in this building, there's about 50 of us, that's what we're focused on.
Daryl, you want to help brewers see the whole picture, don't you?
Yes, we want to help everyone see the whole picture.
See
Like,
the whole picture.
yes.
But in this sense, that metagenomics approach is very much a partnership, based thing. Like you are, it is not something you're just trying to sell people. You're trying to, be problem solving partner along the way.
Yeah, it's true. Like I said, I'm not a salesman, but I believe in all these products. I know how they can help brewers. And like I said, brewing is close to my heart because it's where I started in this whole industry.
Sure, and you know, in that sense, like you know, if you've got an issue, or if you just want to explore what those kinds of preventative opportunities might be, you know, it's easy to reach out, and I think we'll throw out here in the chat some ways to do it. And of course, the last slide that we have here certainly has links to pop back and check out all these ways to get in touch with the BioMeriU team.
Yeah.
Um,
And,
but
and you can, oh, sorry, go ahead.
no, good.
Yeah, and you can always reach out to me directly, and also you can reach out to your sales account manager because we work as a team to make sure we understand your problem and we can help pick the right technology that we think will help solve it for you. So reach out to any of the team, and we will get you to the right place.
And again, we can take some nerdy questions on, that from brewers out there that want to pick Daryl's brain. But you also want to share something that is pretty fresh. In fact, this is kind of a preview of what you all are going to be rolling out very soon into the market for brewers to kind of add more power, helping them see even more of that whole picture. And take some of that control into their own hands. Again, this is still fresh and new. There's still not a lot of data around it that is public, but you just wanted to throw a little tease out here.
Yeah, so I alluded to it, before that we want to make this approachable. I think the workflow is approachable, especially for those in the brewery that have been doing this a long time. But on the back end, sometimes can be a little bit nebulous, right? A little bit difficult to digest, especially if you're not a nerdy scientist. So if you want to start taking these kind of complex data to your leadership and ownership, people who aren't technical scientists, you really need a good, simple, and robust way to do that has a big impact. So in the last year or so, we worked to acquire a Brazilian company called Neoprospecta. They are a sequencing company. It is all they do. And they specialize in 16S and ITS metagenomic sequencing. So they have a whole process and pipeline. They run about 25,000 samples a year, which is orders of magnitude more than what I run. So they have a very, very cutting edge operation in Brazil. And if you ever get the chance to go to Florianopolis, Brazil, do it. It's the most beautiful place there. So we got lucky that we get to travel there to go to this place. So what they've developed is a kind of a cutting edge pipeline. So their workflows are unique. They get kind of the most out of it. They can define and identify more microorganisms than when you're using the commercially or academically available pipelines and workflows that are out there. So they have some proprietary front end work. But in terms of presenting this data, what they have is a pipeline and an analysis engine that makes this stuff really easy. So the Smart Biome pipeline and platform will kind of take anyone like me who's just a microbiologist, and it makes them look like a $200,000 a year bioinformatician. It's really that good. It takes all your complex data, turns it into very clear visuals. So on one here, you can see with the bar charts, this is showing a host of samples, and it's showing what each sample contains and the color codes or individual organisms. The platform is really user-friendly, so you can click on one organism on any sample, and it will show you all the other places that it was found in those samples. It's completely searchable for anything you want to do. And then it can take that data, which is also not the most digestible to a non-scientist, and map it onto your plant. So if you look at the bottom left column, what you see is a plant manufacturing plant map. So it will take this data, all the organisms, and put it right on your map. The big advantage to this is, if you are collecting and trying to trend data, most people, including food and pharma, they're using Excel spreadsheets, and Excel spreadsheets are where data goes to die, right? If you're trying to trend, one sample point, you're like, okay, you're looking at cells and going along the year, trying to make a trend out of it. It's pretty much impossible in yeoman's work. No one wants to do it. So we take this data, put it directly on your plant map. So all you have to do is go, oh, here's a red circle and here's the size of it. That's bad or it's good, So it's right there. You can analyze, weeks, months, years of data just by looking at your plant maps. And then the other
That's amazing. That is absolutely amazing. As graphic designer who designs quite a few uh, 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 information graphics and absolutely spends a lot of time in that space of data visualization. I think you're 100% right here that I could show an executive at a company that I had worked for before, a whole bunch of numbers on an Excel spreadsheet. But
Yeah.
man, when you visualize it like this, it allows you to make, it allows you to synthesize that data and understand the context behind it. And I love this idea that it can pinpoint for you where in your production process some of these challenges and issues might be popping up. What a super cool way to build a system.
Yeah, it really is awesome. We're really excited to roll it out. And the team in Brazil, so we're doing basically copy paste. So in my lab, we're replicating it. And then in France, we're doing it for the European community. It really is an amazing system they have. And like I said, spreadsheets are what you get the answer. There's no budget for that with a spreadsheet. When you come with them with a smart biome plant map, that's when you can say, hey, look at this, and then they can understand it. And that's when you can think about getting those preventive budgets. So the kind of the cherry on top here with the smart biome system is risk analysis. So what our friends in Brazil have basically gone industry by industry and looked at the microbes and have assessed a risk to each industry. So, if you find a Lactobacillus brevis in kimchi, it's not going to come up red, right? It's going to be there and it's fine. That's what you expect. You find a Lactobacillus brevis in your fermenter or in a bright beer tank or something, unfortunately, that thing is going to come up red on the speedometer, right? So again, the risk analysis is a really nice visual to take up to leadership and ownership and non-technical scientists. And it really is industry specific. And this is constantly being updated as they acquire new data. So really the concept of smart biome kind of in three pieces. We call them spot, act, prevent. Spot means, hey, this beer is spoiled. This one's not. It was made the same time. Can you analyze them and tell us what spoiled it so we can try and come up with a decision? Do that. That's spot. It's a small project, easy to do, pretty quick. Act is, hey, we found this problem. We think we've narrowed it down to this section of the brewery. Let's do a swabathon in this area of everything we think is risky. And then we'll identify everything there, come up with what we think is a plausible corrective action, and we'll do that. Hopefully it solves the problem. And then prevent is kind of that continuous monitoring. So let's say you did an ACT project. You found something, on your cold side in your fermentation, and you remediated it. But you don't know. How are you going to know? Was that the actual problem, and was our corrective action good? So you over time, every month or every quarter or every 6 months, you take the same samples, we analyze them, and then you look at the risk and you say, okay, did our corrective action reduce our risk profile or reduce our growth or change the growth of a certain organism that we want to see, or did it not? And then so this is where you go back up the chain again to upper management and ownership and say, look, here's the ROI on this project. We show that the quality is going up, the risk is going down, and so we're not going to worry about our stuff once it gets to the supermarket or the beer store.
Every executive understands KPIs and dashboards, and this is
Yeah.
a set of quality KPIs, right? That
Yep.
in a visual dashboard kind of format that gives you some insight and overview of what's going on in the brewery, and and within that kind of microbiome within the brewery itself. Super cool. Is there some sort of time frame for when this will start to be available next year to brewers out there in the marketplace?
Yeah. Is Jermaine still on the call?
We've
No.
got him in the
Okay.
back here. I'm sure he can answer
So
it.
yeah.
If we can't answer that yet, we'll just leave it open ended for
No,
now.
it's okay. Wait, we have a Q1 launch. I just wanted to see if Jermaine wanted to give more details. But yeah, so I've been tasked with having our lab ready by December 31st, which I'm working on right now. And the Brazilian lab has been existing for years. And then on the European side, if there's any Europeans here, they're looking at launching Q2, maybe into Q3 on the European side. But we're already doing projects in Europe, just not fully in-house. So we contract out a little bit and rely on our Brazilian friends a little bit more. But we aim to be self-sufficient Q1 in my lab and then Q2, 3 in France.
Super cool. Super cool. I've said it a whole bunch, but the technology and the opportunities for quality, the ingredients, everything the brewers are using, and the technology that supports brewing great beer is the best that it's ever been, and the kind of tools that you all have built and are now continuing to innovate on and roll out for brewers take that kind of like granularity so much deeper that to help brewers truly understand in a way that they've just never been able to understand what's actually going on within the tanks, within the pipes, within the entire realm that contacts beer ingredients and liquid itself through the brew house. It's pretty phenomenal to see all of this stuff in action these days.
Yeah, the brewers take the science of this so seriously that we're all kind of victims of our own success. It used to be if you controlled your lacto, PDO, and your DEC and Brettanomyces, you were good. That's all you had to worry about, right? But then as we got better and better at things and diversified, and got more and more intent on quality products, you kind of open up yourself and create these niches and other more complex problems that we need more complex tools for. But it's all in the name of producing higher quality products. So it's really exciting.
Well, because the microbiological world is not static, right? Like it's not like it just exists and it's never changing. Those same microorganisms are evolving and developing tolerances and building survival mutations or giving them different kinds of abilities, and they're becoming different things, and it's constantly a constant process. And so in this sense, like, you can't stop doing it just because you think you got there. Everything wants to thrive and survive in the world, right? And all of these organisms want to promote their own existence. And so it's just staying aware of how all of these things are moving, and that kind of entire dynamic system is just incredibly important.
Yeah, definitely. Nature abhors a vacuum, and when we if we give into our narcissism and think that we have problems solved, that's when stuff comes back to bite us.
For sure,
and it is amazing how quickly these things evolve. I mean, in the lab, we make the lacto evolve by doing serial dilutions in higher hops. It's how we keep high hop resistant strains available to study. So it's very easy for these things to evolve.
Oh man, you guys are genetically promoting super lacto in your own laboratory. It sounds dangerous.
Well, yeah, there are some assays that require it. We need to be able to track it, but yeah.
The most hop tolerant lacto. Well, I'm sure there's some brewers out there that would love to take some of that off your hands to try in some of their beers just for fun.
And it's more, it already existed, but over time, if you passage it, it loses its resistance. So we're just trying to maintain it how it was when we received it from a brewery.
Sure.
So yeah, we're not Island of Dr. Moreau creating monsters.
Thankfully, those lactobacillus are easy enough to keep under control. So you're not worrying about them going too rogue here. And of course, all of this applies to non-beer makers as well as beer. And in fact, as we mentioned earlier, in talking about NA beers, in that kind of space, without the alcohol present, some of this understanding is even more important than it is even in the beer space. And of course, it's incredibly important in the beer space itself. Well, I think that kind of brings us to the end of the structured portion of this deep dive. We did have a question where Hunter wanted to get nerdy and ask you how you process a sample to exclude non-viable cell DNA. Definitely a niche question that I'm kind of wondering what he's even asking there.
Oh yeah, sure. So if you're familiar with our PCA assay kits, we have a viability kit add-on, but you can look these things up if you look up PMAX treatment. Basically, you treat with a chemical that cross-links DNA. So you treat it before you smash everything open. So dead cells will be permeable. So this chemical can permeate through the membranes, bind to the DNA, and cross-link it. So then once you smash everything open, this DNA is immobilized. In order to sequence and do PCR, you have to be able to melt the DNA strands apart to amplify them and sequence them. When they're cross-linked, they can't melt, so they're invisible. So that's it. It's pretty simple. It's just a chemical that's permeable for dead cells.
If anyone else has some microbio questions or biome or metagenomics questions, throw them out there in the comments. We are watching them, and they are coming in from whatever platform that you use. I don't see any other fresh ones here But we'll hang on here for a second and see if anyone has. Daryl, are there any other kinds of interesting things you guys have learned in doing some of this testing with metagenomics for brewers out there? There are you have employed this for a number of brewery customers already, and obviously we're not going to name names, out of confidentiality for them. But there are some things you picked up from here and there, learning by doing this testing for brewers.
Yeah, well, can I give you a brewing non beer example?
Sure, of course.
So yeah, so we had a brewing
Fermentation
customer
is
who,
fermentation, Daryl. It's all
yes,
welcome here, no
all
matter
right, cool,
what you're
yeah,
fermenting.
we had a fermentation issue. So, the world of kombucha can be a little bit mysterious and nebulous and potentially risky when you don't know, what your culture is. You don't know what your mother and baby is made out of that you're using. So we had a customer that decided that they would, look at the constituents they thought were important for their kombucha and create a synthetic culture, a mixture of those. So they did that. They were seeing inconsistent quality after repitch. And so, they couldn't figure out, it's a mixture of bacteria and yeast. And if you're plating for bacteria and yeast, you kind of don't know how much of one, how much of the other is there, after you've recollected it from a fermentation. So we did a study with them, collected samples over time. And what we saw was that a couple of their main components of their fermentation mixture, their synthetic SCOBY, were disappearing after Gen 0. So at Gen 1, 2 of the 4 components were going away. So that was why they could not get consistent quality. So something like that can be a very simple fix with metagenomics, is just IDing that relative concentration of each organism. So as long as you can get a signature for each one, you can tell. And with plating, you can't really tell. You're like, okay, it looks like a yeast colony, it looks like a bacteria colony, but that's about all you can get. So metagenomics can really help you with that relative abundance. And that was a very big problem followed or solved by a pretty simple metagenomics project. And this is the thing where the ROI is immense when a project is in the hundreds to thousands of dollars, and then your impact is tens or hundreds of thousands of dollars.
For sure. Any more questions before we wrap this up? I don't see any in the chat. We've given everyone an opportunity. And anyway, so thank you so much, Daryl, for sharing this perspective on understanding your biome and microbiome in your brewery. And then, of course, using metagenomics to get an even deeper understanding to see the whole picture, right? The whole picture. And, and then, of course, before we go, if you want to see that whole picture, you can follow the links that are out here in the comments in the chat. You can snap a picture of this QR code on your phone and take it right there, take you right to them. And let's see, it looks like, we actually, we had a homebrewer question out there, Daryl, before we wrap it up. For a homebrewer with access to a PCR machine and a DNA sequencer, because we have homebrewers out there like that, is there a kit for them to test their own ferments?
So for not a lot of investment, you could do PCR testing if you had a reason to do that on your home brews. Sequencing is not so approachable. There used to be a machine that was around $20,000, but they've discontinued that. So kind of your cheapest aluminum machine is going to be outside the range, I think, of a home brewer. A long read sequencer from Oxford Nanopore could be in the range, but the accessories that you need and the kind of cleanliness are things typically not seen, even in a small craft brew house. So a home brew, it's probably, a stretch, but that's not to say you couldn't take the samples and send into a third-party testing lab. But if the person wants to contact me, I can give, more details, but it's still a little bit unapproachable, like to that, that level, to home brew and even small craft brew. It's still a little bit of a stretch.
Sure. Well, if you've got more questions, reach out to Daryl directly, reach out to your rep at bioMerieux. Click on the links that are shared out here in the comments, take a picture of that QR code and follow it. And you can learn more about the microbiome in your own brewery. Daryl, thanks. I think we'll bring that to a close. Thanks for everybody who is joining us out there live. And if you weren't watching us live, then thank you for tuning in and watching the recording of this out there on all of the platforms. Daryl, it's been great talking with you. Cheers.
Yeah, thanks a lot, Jimmy, and I appreciate everyone's attention. I know how busy everyone is in the middle of the day, so thank you for joining in.
Let's help make everybody make better beer.
Yeah. All right. Have a great rest of your day, everyone.